DNA Replication by April, Ivana, Mindy, Natty, and Taylor.
An overview of replication. So first, the DNA double helix is unwound to separate the two template strands and make them available for new base pairing. Then, as new nucleotides form complementary base pairs with template DNA, they are covalently linked together by physodiester bonds, forming a polymer whose base sequence is complementary to the base in the template strand. The template DNA is read in the 3 to 5 direction. Now let's go in depth.
Semi-conservative replication is the preferred DNA synthesis method in which two original strands separate and each acts as a template for a new complementary strand. So here's the original strand, and then after it breaks off, it connects with the new strands, which are the blue ones. A template is a structure that in some direct physical processes can cause the patterning of a second similar structure or a molecule that provides the structural mold to create similar substances. So this is a template strand and then the growing strand. DNA polymerase is an enzyme that catalyzes the polymerization of deoxyribonucleotides into a DNA strand. In DNA replication, the polymerase reads an intact DNA strand as a template and uses it to synthesize the new strand. So this is the DNA polymerase enzyme in the DNA strand. Origin of replication. DNA replication begins with the binding of a large protein complex, the pre-replication complex, to a specific site on the DNA molecule. All chromosomes have at least one region called the origin of replication, ORI, to which the pre-replication complex binds. This is an example of a prokaryotic and a eukaryotic origin of replication. So this slide talks about the 3 to 5 direction on the DNA structure. So one key feature on the DNA structure is that it is anti-parallel. So each strand is built in the 3 to 5 direction, but two strands run in the opposite direction, like right here to here and then here to here. And so at one end of the double-stranded molecule, there is a 3-OH exposed on the deoxyribose sugar of one strand and a 5 on the phosphate on the other. Replication forks. Once the pre-replication complex binds to it, the DNA unwinds and replication proceeds in both directions around the circle, forming two replication forks. So this is the pre-replication complex, and then it goes into two replication forks.
So in DNA replication, the primer is a strand of nucleic acid that serves as a starting point for DNA synthesis. So here's the primer, and it's going to be the starting point for this, which is the new DNA being synthesized. So next is primase, which is an enzyme that synthesizes short RNA sequences, or the primers from the last slide. So here's the primase, and it's going to synthesize these primers, which are in red. And then we have the leading strand, which is the strand of DNA being replicated continuously in the 5 to 3 direction. So here's the leading strand, and it's going to keep replicating.
What contrasts with the leading strand is the lagging strand, which is the daughter strand that is synthesized in discontinuous stretches, backwards and shorter. So it must be synthesized differently because it grows in the direction away from the replication fork. So these discontinuous stretches are synthesized just as the leading strand is by the addition of nucleotides one at a time to the 3 end, but grows away from the replication fork. So this is the leading strand growing forwards, and this is the lagging strand, which is synthesized as Okazaki fragments backwards. And here are the different gaps showing that it's discontinuous. Okay, Okazaki fragments are newly formed DNA making up the lagging strand in DNA replication. DNA ligase links the Okazaki fragments together to give continuous strand. So here is one Okazaki fragment and the other, and there's a gap in between. Okay, DNA ligase is an enzyme that catalyzes the formation of the bond linking fragments and making the lagging strand whole. So for this to occur, first the primase forms as an RNA primer. so that DNA polymerase can add nucleotides to the new Okazaki fragments at the 3 end, like right here. And then from there, DNA polymerase hydrolyzes the primer and replaces it with DNA, as in right here. And finally, the last step is that DNA ligase will catalyze the formation of the phosphodiester linkage that finally joins the 2 Okazaki fragments together, as in here, making it one whole strand instead of different segments.
So we have telomeres, and the same way aglets are used to protect the end of our shoelace, telomeres are designed to protect our cells from damage. They're located at the end of our chromosomes and wear out over time, leaving our cells vulnerable to damage, therefore causing aging. So here we have one of our chromosomes, and at each end is a telomere. Then we have phosphodiester bond. It is a bond between a 2-sugar group and a phosphate group that holds together chains of RNA and DNA. So we have our phosphate groups, and they're bonded to the sugar groups.
And that's how DNA is replicated.